And welcome everyone to the fifth and final day of the 2022 annual meeting of the Washington State Society of American Foresters. I'd like to thank everyone for being here. Thank our speakers today, as well as everyone participating. And of course, I want to thank our sponsors for making this happen, as well as the Planning Committee and the Northwest Office. Pretty excited for today. Our theme is wildlife and ecology. So we've got a couple of speakers plus our last two presentations from Green River students. Our first speaker today is Ray Davis from the Forest Service. He's going to speak to us about the past 25 years of monitoring late successional and old growth forests. After him, Malia DeVivo from the Washington Department of Fish and Wildlife is going to talk to us about chronic wasting and other diseases in cervids. And finally, we'll wrap up with two presentations from the Green River students on cover trap surveys and amphibian diversity in natural and urban environments. Just like the other days, if you have questions, please just put them in the chat, and Andrea will ask them at the end during our Q&A with the speakers. Of course, during the Q&A, if you have a question, you can also raise your hand, and Andrea will get to you. Finally, we do have credits today, I believe, Andrea, and there will be a survey at the end of the conference that Andrea will post in the chat. So just keep an eye out for that, and you can follow the link and fill it out to get your credits. Without any further ado, I'm going to introduce Ray Davis. So Ray is the monitoring lead for Old Forest and Northern Spotted Owls at the Northwest Forest Plan Interagency Monitoring Program. He first began with the U.S. Forest Service in 1989 and spent much of his career implementing the Northwest Forest Plan on the ground in the Siuslaw and Umpqua National Forests before transitioning to monitoring. Today, he's here to give us a talk about the last 25 years of that monitoring. So Ray, I'll hand it over to you.
Thank you, Elijah. Hello, everybody. I'm going to share my screen and we'll get into the slides. Thanks for the interest opportunity to speak to you about what's going on with the Northwest Forest Plan Monitoring Program. So I'm going to just give you a quick view of what we've seen in the first quarter century of monitoring old forests. There's other things we monitor like spotted owl populations, habitat, aquatic conditions. Today we'll focus just on the old forest part of the monitoring. And I'm going to start with this slide right here to provide folks who may not be familiar with the area, the Northwest Forest Plan. It's essentially the range of the northern spotted owl in the United States. Spotted owl was listed under the Endangered Species Act in 1990. It's also an area that stands out when you're looking at the lower 48 in terms of above ground forest biomass. There's quite a lot of productive forests in that area, as you guys are probably very familiar with. So the Northwest Forest Plan contains roughly 50 million acres of forest land. So that's what this map right here represents. The monitoring program primarily focuses on what's going on the public forests managed by the federal agencies, mostly by the Forest Service in Region 6 and then Region 5, Northwest California, but also the BLM and the National Park Service. Those are the three primary federal agencies that manage those forests. This next slide shows you a snapshot of what old growth forests looked like around 1940. And you can see that not all the forests were in old growth condition back in the middle part of last century. There's these gray areas represent places where there was a stand replacement fire, high severity fire back in the 1800s. And then pop ahead 50 years, roughly. This marks the year that the Northwest Forest Plan was designed. It was implemented in 1994, but they designed it in 1993 based off of 1993 maps. And you can see this is largely what led to the listing of the northern spotted owl and the Northwest Forest Plan. You'll also notice, if you look closely, some areas such as the Siuslaw National Forest, where I used to work, have actually recruited back into an old forest condition since 1940. So these are those footprints of those old fires that have had enough time to grow back into an old forest condition. So some of that's happening out there in the landscape. Now, the Northwest Forest Plan is a long-term management plan. The expectation was that within about a century, there was land use allocations that were delineated under the Northwest Forest Plan that were designed to maintain and restore old forests into the future to produce a well-connected network of old forests on federal forest land, something that more or less it would look something like this, something more akin to what it looked like prior to the listing of the northern spotted owl. So this was the goalpost. This still is the goalpost for the Northwest Forest Plan. So to monitor old forests, the first thing you got to do is define what is an old growth forest. And there's been lots of efforts at doing that. And it's somewhat challenging to come up with a definition that everybody's satisfied with. What we chose to do for the monitoring program is we focus our definition on the amounts of these 4 different forest structure elements that are commonly associated with old growth definitions. So we have density of large live trees, the density of snags, the amount of down wood on the ground, and the size class diversity within the stands. So multi-story stands versus single-story stands. So in combination, there is an index that was actually conceptualized by Tom Spies and Jerry Franklin back over 30 years ago, called the Old Growth Structure Index. And this is what we've operationalized for our monitoring program. We basically take measurements of those 4 elements in combination. As they start to add up, they represent this continuous gradient of forest succession from a stand replacement event, where OGSI index here on the y-axis is 0. And as the forest age here on the x-axis, as they grow older and older, all those things start to add up, and eventually the curve looks something like that. So once we establish that relationship, that curve, we're now able to pick different thresholds along the OGSI index from 0 to 100. And what we do is we use 80-year thresholds. So we look at the average, OGSI score for 80 plus or 80-year or older stands. And we intercept that curve and we come over here, and that would be the index that represents late successional and old growth. We also want to focus on the old growth part of the spectrum. So we look at the plot data and we come up with a threshold on OGSI that's higher, and it represents that part of the spectrum. So between those 2 things, those are what we monitor acres of through time. So if that's not complicated enough, it turns out we have over a dozen different types of forests within the Northwest Forest Plan area. These 6 right here on the map on the left represent about 90% of the forested area, with Western hemlock and Douglas fir taking up about 50% of that area. So you can see the curves, those OGSI curves, vary by forest type, and the thresholds also vary by forest type. But when we put it all together, we get a pretty continuous looking map here that represents areas that aren't old growth to areas that are really good old growth. And you can see the Olympic Peninsula, the west side of the Olympic Peninsula really stands out range-wide in terms of old growth. So that's the definition part of it. So once we have it defined, then we have to map it. And so the way we do that is we take the forest inventory analysis plots that are remeasured on a 10-year cycle. That's what these green dots over here in this map on the left represent. They're densified on Forest Service lands in Region 6, Oregon and Washington. So that's why the green stands out more in those 2 states. And we relate that plot data to spectral imagery from Landsat time series. So we have a time series going back to 1986 up to 2017, and different types of frequencies of light reflecting off of the forest. We associate that light with other environmental variables like topography, climate, and location, and we impute those plot attributes to the rest of basically every pixel on the map using a gradient nearest neighbor in imputation process. I put a link down here where you can download the 2017 maps for this 3-state region, but Essentially what we produce are annual wall-to-wall maps, now going back to 1986 and currently up to 2017. We'll be updating those to 2021 by the end of this year. So that's the background. These next slides are the story. So that's what monitoring is all about, really, is to tell a story. What's going on? Are we achieving our objectives that we thought we would achieve when the Northwest Forest Plan was designed? And so this is the first 25 years of that story. Again, the plan was designed as a long-term, century-long plan. So within the first quarter century, for the late successional old growth, the map trend indicates more or less a stable trend from 1993 up to 2017. And it's slightly decreasing for the old growth, but still not really significantly decreasing. So more or less what we saw for the first quarter century in terms of late successional old growth forests was a steady state. So, a quarter of a century into this 100-year plan, we haven't really seen a lot of recruitment that is exceeding the amount of losses that we're seeing. I'm going to get to that here in a second. So that's the story. We're basically maintaining what we had as of 1993. And then we compare those map estimates, by the way, to the plot remeasurement periods, just to make sure we're more or less within the error bounds of what the plot estimates are. So in addition to the trend line, we want to know kind of where things are changing. And so what we do is we take these maps, these bookend maps is what we call them. So here we're looking at the 1993 bookend for OGSI 80, again, late successional and old growth forest. And on the right, we see the 2017 bookend map. And we just summarize all those pixels into these 190,000 acre hexagons. It makes it easier to kind of see the patterns. So, the darker the green up to this light dark blue right here represents more OGSI 80 within those hexagons. And so here's what it looked like in 1993. Here's what it looked like in 2017. And then we just difference those two maps so we can see how things are changing. Are they improving or are they going the other direction? And what this map shows is along the coastal margins where forests are very productive. That's where we're seeing our biggest gains in that first quarter century. And then as you'll see here in a subsequent slide, most of our losses, which are in the shades of orange and red, are happening in places where we've seen quite a lot of wildfires. And this is the same picture, except this is the old growth part of the spectrum. So you see a lot less acres of old growth. Again, the Olympic Peninsula really stands out as having a lot of old growth on the west side of the peninsula. And the pattern of change is more or less the same, with some increases here along the coast, mostly in coast of Oregon, and some decreases where we've seen fires. And while things are increasing along the coast of Oregon, there's still not a whole lot of old growth in terms of percentage of a 190,000 acre hexagon. So we still have low amounts, but the trend is going in the right direction. And so in addition to what's happening with, is it going up, is it going down, we want to understand what are the causes for those changes. So we also, in addition to mapping the forest, we map the forest disturbances. And we partner with Rocky Mountain Research Station on this effort. And we produce these 30-meter pixel maps on an annual basis of timber harvesting and wildfires. We also do insect and disease, and we summarize those maps by the same 190,000 acre hexagons. And so here's the pattern of timber harvest, the pattern of wildfires, and the pattern of insect and disease. And in terms of losses to old forest, on federal lands, the harvesting accounted for about 250,000 acre of loss between 1993 and 2017. Wildfire exceeded that greatly, about 800,000 acres of loss from wildfire, and mostly on federal. Well, all of this actually is on federal. These losses are on federal forest right here. And then insect disease is mostly showing up in those droughty parts of the Northwest Forest Plan, the east side of the Cascades, down in the Klamath region in Northwest California. And it only counted for a very small percentage or acreage of loss. It's more, it's a lower magnitude, kind of a chronic, slow process of a disturbance as opposed to a fast, high, disturbance that you see with the fire or timber harvest. So that's what's causing the changes. The last thing we want to know is, are we achieving some of the objectives outlined in the Northwest Forest Plan? And one of those objectives was focused on the abundance of late successional old growth forests, or OGSI 80, and trying to get to with what they determined over 30 years ago was a natural range of variation. So this, above six, in terms of, this map shows you the proportion of the federal land covered by large patches of old forest exceeding 1,000 acres in size. And so we want to get the hexagons into the green or blue area colors here, and get out of the pinks and the yellows. And we can see we've made some progress in 25 years. That's what the blue hexagons here on the right map show. We made some progress to achieving that objective. We still got a long ways to go. And there are some places where it's gone down, but mostly we've made progress in terms of the abundance of late successional old growth forest on federal lands for the first quarter century. We also look at connectivity, and I think that's one of the focuses of this week's meeting. And so here are some maps of the connectivity as measured by the average distance between those large patches, 1,000-acre plus patches of old forest. And so you can see that mostly within the estimated natural range on federal lands, but along the edges of those and the Coast Range of Oregon, we're below that. We've made some progress here in the Coast Range. So again, the blue means we're going in the right direction. The red means we're going in the opposite direction. And you see a lot more red in this map that corresponds to those large wildfires we're getting on federal lands. And so large wildfires have been challenging some of these expected outcomes. We're starting to see that in this first quarter century of monitoring, but we still haven't have been able to maintain. So in spite of the losses, they amount to about a little over 1 million acres of losses. There's been enough forest recruitment in that same time frame to more or less kind of balance the sheet and things out. So again, we've maintained it. That's what the story says. In spite of 1 million acres of loss, we've had about roughly the same amount of recruitment from some of those old fires from the 1800s and other things. And so at the end of 25 years, this is the story. But when you're monitoring things for long enough, we can start looking at these trend lines, and we can start also thinking about the future. So I have an epilogue here. Because of the increasing rate of large wildfires on federal lands, so since the end of this monitoring report, which was middle part of 2017, we've had 4 really big fire seasons represented down here with well over 1 million acres burning in 4 of those years. And then in 2020, you're familiar with those fires. That was an exceptionally large fire season. And so what we're seeing is the story is changing. And so monitoring cycle has been every 5 years, but the frequency of these large fires is starting to push us to accelerate the frequency of our monitoring to an annual frequency, because by the time we do 5 years of, another 5 years of monitoring and go through the process of publishing those reports and everything, the story's already changed as exhibited in these 2 slides. This is a story that's going to be coming out in the 25-year monitoring reports. Everything looks okay. And then this is the situation today. So we're concerned about this declining trend that we're starting to see as a result of increasing annual area burnt by wildfires every year. And we're also looking at the climate science data. And so these maps right here, we published these and actually 10 years ago, and we updated them for climate change 5 years ago. These are maps that represent what we call the normal fire environment. So essentially, red areas here mean that within a climate normal period, 3-decade time period, you're going to get a lot of fires in those red areas. And in the blue areas, they burn too, but they burn infrequently. And so this is what it looked like as of today. And then jump ahead 20 years, a little bit more reds. And by the end of the century, you can see there's a lot more area within the Northwest Forest Plan that the climate science indicates are going to be burning a lot more than they are burning today. So what we consider normal today, and this is a map of the last half century of fires, you can see that's kind of the areas in the Northwest Forest Plan that we think burn, because that's what we've observed. And you can see how well those fit that model, that map. This is last year's fires, over 1 million acres burnt. And so what we anticipate is these red areas just start to fill up year after year with large wildfires. And as climate change keeps heading in the direction it is heading, then there's going to be places even on the west side of the Cascades in Oregon, even up on the Gifford Pinchot in Washington, and that are where large wildfires are going to just be more commonplace. So that's where things are going. And so we're going to be monitoring the situation closely in terms of the forest trends, and we'll just see how things go. So with that, I want to acknowledge my partners in this work. So Dave Bell from PNW Research Station and Matt Gregory from Oregon State University. Those guys are instrumental in making those GNN forest maps. Andy Gray is PNW Research Station, and he's instrumental in the FIA plot analysis and support. Trishan Young and Sean Haley work for Rocky Mountain Research Station, and they are instrumental in mapping the forest disturbances, such as timber harvest, wildfire, insect, and disease. And then Andrew Stratton is Region 6 Forest Service, and he provides a lot of the GIS support. So with that, I'll open it up to any questions folks may have. I don't see the chat box, and I guess, Andrew, you want me to, or Elijah, you want me to stop sharing my screen?
No, it's okay. We might want to go back if people have questions about the maps. And so looking at, as you showed the maps, the wildfires. So is this going to prompt any maybe changes in management around managing for old growth, or, you know, in the area, in the stands adjacent to it, or?
I guess that remains to be seen. So most of the focus right now is in places that we are familiar with burning a lot. So the east side, the Cascades, and the Klamath. And that makes sense. There's a lot of communities at risk, but this is fairly new information. I think it takes a while for information to kind of sink in and be accepted. We are talking about this. Folks I work with are talking about this. And in terms of management, I also monitor, I oversee the Spotted Owl monitoring program. And I kind of am starting to use an analogy with barred owls and these maps that we're looking at right here, because in 1993, when we started the monitoring program, we knew that there were barred owls starting to make their way into this area in the northern spotted owls range. They were going from north to south, and I think we lack the imagination of what that might mean to the northern spotted owl populations. And by the time we figured out what it meant, they had already made it all the way down into California, and were starting to make their way into the California spotted owls range on the Sierra Nevadas. So, you know, the spotted owls are the trends are really bad, especially up in Washington. They're getting pretty close to the local extinction points in some areas, and it's largely because of barred owls. So I use that as an analogy because I see fire sort of invading in the opposite direction. So this is where we're used to, but it's slowly starting to creep north and west. And I think, you know, we're talking about managing, maintaining, and restoring old forests. It's a long-term prospect. You just don't do that in a few years. It takes a century to do that. So we should be thinking in terms of a century. And so looking ahead to the end of this century, this should be something on our minds, I think, and not just for Forest Service. I think all the forest management agencies should be thinking about this change in terms of the way they manage the forest. And so but it's fairly new information, and we'll see. we'll see what happens over the course of the next few decades.
Nice. And how much of that will also need to bring in industry partners who have timberland adjacent to the federal, to the Forest Service or state agencies or federal agencies to make them part of that discussion, showing where the wildfire situation might be in 100 years?
Well, I know that one of the co-authors of this paper was Andrew Yost, and he's the Oregon Department of Forestry forest ecologist. And so he's been talking down here to the state foresters and in Oregon, and I don't know to what extent the non-public agency, like private industry, I don't know, but these are publications that are out on, available for people to download and to become familiar with and read. But to my knowledge, there has not been a whole lot of outreach or interest in terms of these particular maps.
Mm-hmm. Nice. And so you mentioned eventually trying to move towards annual mapping. So kind of what are the underlying technologies that are going to be enabling you to move that direction? Because I imagine that's kind of going to be a game changer in terms of monitoring.
Yeah, it is. It's going to be a huge game changer. But fortunately, one of the things that we've been able to do is keep up with technology, which has really advanced since we started doing this. So there's a Google Earth Engine cloud computing platform where all this Landsat imagery resides up in the cloud. And so we are working with Google Earth Engine, and we actually have, they call them Earth Engine applications. And so we haven't quite implemented it for GNN, for the forest vegetation and structure maps, but I would say we're within a year's, we should be able to take the mapping process that I quickly went through and basically grab the latest Landsat imagery, which gets automatically cleaned and updated into the Google Cloud, and then basically apply these mapping algorithms to that, so that it almost becomes a push button automated process at some point, where you can just go to and see what the latest trend is. We actually have that implemented for our northern spotted owl habitat maps. And we're working on that for the marbled murrelet maps right now. And I actually have it implemented for the old growth maps, which are a derivative from the GNN maps. Once we derive those maps, then we have a process where we use disturbance severity to update those on an annual basis. And so, so yeah, those applications currently exist, and we're still kind of developing them. And but we have some we could share if folks are interested, that will be updating on an annual basis. So you'll be able to kind of see how these trends change. It allows you to pick different parts of the Northwest Forest Plan, such as federal lands or particular forest, and it'll give you the same trends for those areas.
Nice, thank you. All right, looks like Elijah, you have your hand up.
Yeah, so right on the page with the probably answer next slide.
Hey, Elijah, I can't understand.
Oh, let me take a look. Your next slide, I think it has the graph hub on it. Yeah.
Elijah, you were coming in very low. Maybe you can type your question in the chat. Andrew can read it.
Yeah. Okay, so it looks like, so Eliza's question was, what are the numbers behind the burn frequency map? Is it probability of ignition? Okay.
Yeah, these are fairly simple models that look at climate. So fire season average, or no, maximum temperature during the fire season, the average precipitation for the head and shoulders of the fire season, so from about May to the end of October, And then topography, like slope and elevation. So very simple kind of intrinsic parts of what constitutes the fire environment. And we trained these models, we trained them on fires that burnt from 1970 to 2000. And then we extrapolated the model we basically make a model for every climate normal. So this one right here is the 1990 to 2020 climate normal. This one is 2010 to 2040 climate normal. Again, climate normals are 30-year averages basically of weather data. And then for the projections into the future, we use NASA downscaled Earth exchange climate prediction data for precipitation and for temperature.
Okay. All right. So it looks like one question came in. So is the Colville, Umatilla, and sorry, I'm going to mispronounce this really badly, Malheur and Walla Walla Whitman not included in the Northwest Forest Plan extent?
Yeah, so the Northwest Forest Plan is mostly west of the Cascades, but we have a little bit of the East Cascades as far as the spotted owl goes, which is basically the edge of the Douglas fir, where you find the Douglas fir type forest, grand fir type forest. Once it turns into more pine type forest, spotted owls don't use those types of forests. And so the Northwest Forest Plan ends on the east side of the Cascades and doesn't include those forests. Now the model, that if you're interested in this map right here, we do have that for the entire state of Oregon and Washington. Mm-hmm. And there's a publication. This is the publication. Here's a link to the publication. And we also have one of those Earth Engine apps that allows you to kind of see what's going on those forests in terms of the change through time.
Nice. And so for you personally, how do you feel optimistic even seeing these declines, or are you concerned? Or kind of how do you stay focused on knowing we're trying to make a difference here with increasing old growth when we have climate change and this, the barred owls coming in?
Well, I'll stick with the old growth and not get too far into the barred owl issue. That's a different module. But for the old growth, I mean, we actually have a pretty good history, at least on federal lands, of all the regeneration harvesting, all the clear cutting that has happened. And it just takes time and lack of disturbance. So if those plantations that were harvested, let's say in 1960, there was quite a lot of clear cutting in 1960 and 1970. So give them about 80 years. We're looking at 2040, 2050. And we're anticipating once we get to those decades, if those plantations have not burned up and been reset back down to an early sterile condition, we anticipate about 2040, 2050 to see a pulse, an increasing pulse. And so stay tuned for that, but that's kind of the expectation. Obviously, some of those plantations will burn up at high severity. Some of them won't. So there will be some uptick here eventually. Now, will it be a big enough uptick to offset what we're currently losing to this increasing footprint of fire remains to be seen. We'll I mean, I never give up hope. I always try to maintain optimism. And we'll just see. I think, we have the science and the technology and the tools and the people to kind of deal with this. And so we'll see what happens.
Nice. All right. Well, thank you, Ray. Is there any other last questions? If so, feel free to post them in the chat or raise your hand. All right. Well, if something occurs to you later on, feel free to post in the chat and we can circulate to Ray later. Thanks.
All right. Well, thank you, Ray. Andrew, can you guys hear me? Okay. Sure. Thanks, Ray, for the talk. I really appreciate it. I'm going to introduce our next speaker now, Dr. Malia DeVivo. She's an ungulate research scientist for the Washington Department of Fish and Wildlife. And for the last 5 years, she's been working on predator-prey relationship projects specific to Eastern Washington deer and elk populations. She's also worked on the recently adapted state chronic wasting disease management plan. and assists with surveillance efforts related to chronic wasting disease and other hemorrhagic diseases in cervids. Today, she's here to talk to us about those diseases. So, Malia, if you're ready, I'll pass it over to you.
Hello, everyone. Do you see my screen? Perfect. Let me rearrange all of my screens now. Okay, let's see here. Okay, I think I'm set. Well, first off, I just want to thank you all for attending and inviting me to speak about chronic wasting disease and hemorrhagic diseases of cervids. Again, my name is Malia DeVivo. I am the ungulate research scientist for Washington Department of Fish and Wildlife. My job duties do extend statewide, but I reside in Spokane, Washington on the east side of the state. And so far, my work has only occurred on the east side of the Cascades, and I haven't been fortunate enough yet to work on the west side of the Cascades yet. So I've been in this position for 5 years now, a little over 5 years now. And just a really brief background about myself and why I'm here talking to you about diseases. I have done primary research on chronic wasting disease and its impacts on mule deer, specifically in Wyoming, where CWD is endemic. I have not done any primary research on hemorrhagic diseases. However, we did experience an outbreak, which I'll talk about here during this presentation, of EHD and blue tongue last year. And so I assisted our state wildlife vet and our wildlife health staff to monitor that disease. And so I can speak about the disease and that particular outbreak. So first off, I just want to clarify what we're talking about today and what are hemorrhagic diseases of deer in Washington. There are actually 3 different types of viruses that circulate in Washington deer, and all 3 viruses cause hemorrhagic disease. So there's adenovirus, which is found in Western Washington, so on the west side of the Cascades. This virus belongs to a small group of viruses that can infect a variety of animals, both wild and domestic. AHD or adenovirus in deer was first identified in California in 1994. And since then, it's been quite common throughout California and Oregon. And so because of that, it has most likely afflicted Washington deer in the past. But we actually did not first document adenovirus in Washington until 2017. Adenovirus in particular really affects our black-tailed deer populations, and it primarily, it can infect both adults and fawns, but fawns tend to, we see the highest mortality in that age class with adenovirus. Today, I will primarily focus on 2 viruses, blue tongue virus and epizootic hemorrhagic disease virus. Both of these viruses also cause hemorrhagic disease in our ungulate species in Washington. These 2 viruses circulate primarily in eastern Washington, so east of the Cascades. And so for the purposes of this talk, I'll be talking about those 2 viruses and the hemorrhagic disease that's caused by blue tongue and EHD. Just wanted to clarify, when we talk about HD in Washington, it is caused by 3 different viruses. So today I'm actually going to talk about 2 diseases that affect our even-toed ungulates in Washington. And so the first disease that I'll introduce is chronic wasting disease. And so that disease is specific to the family Cervidae. So those species within the deer family. So in the wild, susceptible hosts include moose, mule deer, white-tailed deer, elk, and caribou. And then EHD and blue tongue actually spans 3 different families within the ungulate order. So EHD and blue tongue can infect members within the family Cervidae as well as Bovidae. And then it also affects pronghorn antelope. So it has a much wider host range than CWD. I'll also point out that EHD and blue tongue can be pretty significant in domestic ungulates. So when we see outbreaks in wildlife, sometimes we will also document cases in domestic cattle and sheep. So what causes these diseases? So for the next several slides, I'll present, I'll speak about CWD and blue tongue EHD in tandem, so you can see the differences between these 2 diseases. So first, CWD is caused by a prion protein. What is unique about this agent of disease is that it contains no nucleic acid. So it's not your typical virus or bacteria that causes disease. It's actually a protein. These prion proteins are ubiquitous among mammalian species. So in fact, humans, all of us have the prion protein. What causes disease, though, is that the normal prion protein misfolds, and this misfolding of that prion protein causes disease within the susceptible host. And I'll also point out, because this protein originates within ourselves, diseases that are caused by prion proteins are unique in that the infected host does not amount to an immune response, because the body recognizes it as its self proteins. So that's what makes these diseases even more interesting and even more complicated to deal with. EHD and blue tongue virus is in the name. It's caused by a virus. So this is a very standard disease-causing agent, just so it does have nucleic acid. And because it does have nucleic acid and it's an exogenous agent that causes the disease, the host species can mount to an immune response, which is important to know, because with outbreaks and large outbreaks of EHD virus or blue tongue virus, after a large outbreak, we can see a population that survives an outbreak that are actually immune to subsequent outbreaks of either one of the viruses. So that's important when we're talking about how frequent do these viruses circulate, and then immunity built up in those herds that are affected. So let's talk a little bit about the history and distribution of each disease. Chronic wasting disease was first documented as a syndrome of captive mule deer in Colorado in the late 1960s. Here I'm saying syndrome because at that time they didn't know it was a prion disease. And so these captive mule deer, and actually black-tailed deer as well, were in this facility for a research study looking at the effects of nutrition. And so at that time, they were documenting high mortality of this wasting syndrome that was occurring in those research animals. And then it wasn't until the 1980s when Dr. Beth Williams identified it as a prion disease and then named it chronic wasting disease. And then shortly after that discovery, they started to detect it in wild populations of deer and elk in Colorado and Wyoming. So at this time, 28 states and 4 Canadian provinces have detected CWD in North America. Unfortunately, last year was the first time that Idaho had their first detection of CWD in their wild herds. And so unfortunately, that puts CWD, known cases of it, only about 50 miles from our southeast border of our state. So it just keeps creeping closer and closer each year. So it's of great concern to all of us. EHD and blue tongue, as you can see, has a much wider distribution in the US. There were accounts of periodic outbreak outbreaks in southeastern US. And again, during that time, they didn't know it was EHDV at that time, but they described this ailment in deer that caused what they called it black tongue. But so in the late 1800s, there's accounts of black tongue in deer, which later was discovered to be actually epizootic hemorrhagic disease virus. Blue tongue virus was first confirmed in Texas and California in the 1940s and 50s. And so currently the distribution is this southeast to northwest band of hemorrhagic disease in deer. So let's narrow down the distribution now to Washington. So first, CWD, if we zoom in on that larger map that I just showed you, fortunately for us, we have not detected CWD in Washington state. And if you notice, I'm saying we haven't detected it, and that's because it could be here, hopefully not, but through surveillance and all of the tests that we have run on animals that we've collected samples from, none of those have come back positive for CWD. And so I'll also talk at the end about our surveillance program and how we conduct surveillance to make sure that it is not in our herds currently. With EHD and blue tongue, again, we just most recently had an outbreak last year. So last late summer, early fall, we started to get reports of sick or dead white-tailed deer all throughout Eastern Washington. And so I'm just showing the distribution of those reports throughout Eastern Washington that we got in 2021. We have had outbreaks on the east slopes of the Cascades, but what I didn't mention was that this hemorrhagic disease is actually considered the most important viral disease of white-tailed deer. And since in Eastern Washington and where you see that distribution, it's primarily a white-tailed deer system, especially in the Northeast. And that's why we see a lot more reports of dead deer in those regions. White-tailed deer, when they're infected, we can see pretty high mortality in that species. Mule deer, they can die, and we have documented dead mule deer, in fact, just last year, but many of them also can survive. They do get sick, and they can have secondary effects to the disease, but it seems to be more of a morbidity than rather high mortality in mule deer. Okay, so let's talk about transmission. So CWD can be transmitted year-round, and there are certain activities that can increase transmission, and that's because deer and elk with CWD can live months to years with the disease. And unfortunately, during that time, they're shedding infectious prion proteins, either through their saliva, urine, and feces. And then also when those animals eventually succumb to the disease, those carcasses on the landscape are contaminating the environment. So transmission for CWD not only can occur directly from a sick individual to a susceptible individual, but also indirectly when they're shedding those prions or the decaying carcasses on the landscape are creating these areas of transmission. Oh, and I did mention that certain activities can increase transmission. So because you have this direct and indirect transmission route, areas where animals are artificially congregated, or you have really high concentrations and potentially high contact rates, such as this picture is showing a salt lick out on the landscape. These are areas that we consider hotspots for transmission, especially for CWD. There's been recent research that has shown that CWD prions actually stick really well to salt licks. There's something about the salt lick that allows those prions to attach to that material. And then unfortunately, other deer that come to that salt lick, they're licking the same spots, and then they're becoming infected. All right, so EHD and blue tongue, is quite different than CWD. So the transmission of EHD virus and blue tongue virus is supported through a biological vector. So you might have heard of biting midges, or they're also called no-see-ums, but they're an insect that's within the genus Culicoides. And so the transmission of these two viruses occurs through a vector. So because of that, that vector is not transmitting disease year round, because the vector is only out at certain times of the year. And that is primarily late summer, early fall. So there's an environmental component to the transmission of this disease. So, and that's why we see outbreaks when the vector is most prominent on the landscape. And so that's going to be that late summer, early fall season. The good part about transmission with this virus is that because it has to go through the vector, with the first hard frost, the vector is killed, and so then transmission ceases, and we see a decline in mortality among susceptible animals. So it is very seasonal, and die-offs don't occur year round. I will mention that there are certain activities with these viruses that can increase infection rates. So if deer are congregated around certain water sources where the vector is prevalent, and usually that's why we see large outbreaks during really dry, hot summers, because deer are now congregating over the limited water resources. And so there's more deer in risky areas where they can become infected. All right, so pathogenesis, which basically means how does this disease develop in the animal? With CWD, this is a disease of the central nervous system. So it causes holes in the brain. So again, the prion protein, it misfolds, and unfortunately that misfolding of the prion protein, your body isn't able to eliminate damaged prions like it should normally. And so those prions don't break down, they build up in tissues like the brain, and then the body tries to get rid of them, but unfortunately it ends up killing normal tissue. And so when you look at a brain under a microscope, it has a bunch of holes in it. And that's why CWD and other prion diseases are grouped into a family of diseases called transmissible spongiform encephalopathies, because the spongiform part of that name describes kind of the spongy look of the brain material under the microscope. With EHD and blue tongue, this is a disease of small blood vessels. So that's why it's called it causes hemorrhagic disease. So basically, all the small blood vessels in the infected animal become leaky and cause hemorrhage. And you can also, the animal will also get some ulcerations in their mouth and upper GI tract. We can see necrosis of their muscle tissue. And oftentimes too, whenever we open these animals up, we see pulmonary edema, which just basically means their lungs are fluid filled and primarily with blood. So essentially these animals bleed out internally. Clinical signs. So what do sick animals look like when they're infected with these diseases? So with CWD, if an animal is able to get to the clinical stages of disease, they'll often look emaciated, they're drooling, they're uncoordinated. If they're a male and they still have their antlers, a lot of times those antlers are very brittle and sometimes they've retained the velvet. I will say though, that the majority of animals that are infected with CWD look absolutely normal. So like I said, this disease takes months to years to actually cause death in the animal. And until the last few weeks to months where the animal is potentially going to succumb to disease, that animal is going to look absolutely normal. So there's a lot of animal, if you have really high prevalence of CWD in your herd, the majority of them are going to look normal. And so that's why we really, really stress to have a surveillance program, and that for individuals who harvest deer or elk should really get their animals tested, because there's no way of really knowing if your animal is infected unless you have it tested. With EHD and blue tongue, infected animals will be lethargic, disoriented. Many times we get calls about deer foaming at the mouth, and sometimes it looks bloody. As the blue tongue virus name suggests, they sometimes have a blue tinged tongue, or back in the 1800s, like I described, they described it as black tongue. So again, because it's a disease of the small blood vessels, it's just constricting those vessels and causing them to leak out into tissue, causing blood to leak out into tissues where it shouldn't be. Unlike CWD, when animals die of it, then they look terrible. With EHD and blue tongue, dead animals will actually look like they're in pretty good condition. So this disease is not chronic. In white-tailed deer, if they succumb to it after infection, it potentially only takes days for them to die of the disease. And so I have a picture there in the middle of we had white-tailed deer collared in Northeast Washington. We experienced this outbreak last year. We had several deer die. And so this deer, if you look at her, she looks in very good condition, and actually she was. And so basically what's happening is they're not able to actually live long enough to experience any sort of side effects where they would lose body condition. So typically what we get calls about are these really healthy looking deer just basically tipping over dead. So diagnostics, so again, those clinical signs don't tell us specifically that that animal has the disease, so we have to run tests on tissues. And so with CWD, there are various tissues we can collect. All of those tissues are located somewhere in the neck or head of the animal. So you'll often hear us ask hunters or people who salvage roadkill deer or elk to save the head, because the tissues we need to sample are in the head. There are several different tests we run, and those tests can then tell us for sure if that animal was infected with CWD. With EHD and blue tongue, this is a virus, so the virus replicates and primarily in bone marrow. So when we do have outbreaks, we ask the public to report dead deer, and we'll go out and collect usually just a long bone, so usually the femur, and within that femur we'll collect the bone marrow, and we can run tests to replicate or amplify the virus and detect if that animal actually had an active infection. We also sometimes take lung, and if you remember that really nasty picture of that lung, basically what we're looking for are the cellular destruction associated with hemorrhagic disease. And so these are just various tissues that we collect to actually confirm that these animals had either one of these diseases. All right, so impacts to wildlife. With CWD, this is 100% fatal disease. There's no vaccine or treatment. So if you can imagine with information like that, you would suspect that potentially this does have an impact on wild populations. And in fact, unfortunately, it can. So we have documented that CWD was associated with population declines in both deer and elk in the endemic areas around Colorado and Wyoming. So in elk, if CWD is able to get as high as 13% in the population, they documented declines in those populations of elk. Very similar with white-tailed deer and mule deer. The mule deer population that I studied in Wyoming, we actually saw that if prevalence is at like 30% in the females, which is incredibly high. Unfortunately, that can impact the population, and we saw an annual decline of about 21%. So pretty big impacts in those populations, unfortunately. It is now being recognized. So when we did those studies, previous to that, it really there wasn't much known about how do we actually tackle CWD in wild populations. And so for many states, including Colorado and Wyoming, not much had been done other than doing surveillance. So tracking the disease, how prevalent was it in the populations, where was it occurring, where was it spreading to. Now, unfortunately, we know a lot more about how the disease is transmitted and potential ways we can manage for the disease. So I'll talk a little bit about that in the next slide. With EHD and blue tongue, mortality and morbidity depends on not only the virus, the specific virus that's circulating. So oftentimes with outbreaks, it's associated with one or the other virus. So some years it's EHD, some years it's blue tongue. Curiously, our state wildlife vet pointed out to me that it was kind of odd because last year's outbreak was associated with both viruses. So we were actually detecting both viruses circulating. And actually some of the animals we tested actually were infected with both viruses within the same host. So I was told that that's quite rare when we have outbreaks. It's usually one or the other virus. So it also the outbreak depends on the host species that the virus is infecting. So again, this is really truly a disease of white-tailed deer. Mule deer can get sick. Other states have seen die-offs of pronghorn when blue tongue is circulating. Bighorn sheep can be affected as well. So it depends on the host species. Elk, not so much. We don't really see mortality. They do get infected and they can mount an immune response, but for the most part, they survive. Again, these viruses don't have a vaccine. We don't have a vaccine or a treatment for these animals. And so we really don't have a clear understanding the long-term impacts of hemorrhagic disease in the wild. We do appreciate the short-term losses that we do see. So again, last year, we were fortunate enough to have collared animals in the areas where we saw this outbreak. So in Northeast Washington, we had collared white-tailed deer. And so by monitoring those animals, we saw about a 25% loss in the collared animals. With mule deer in the Blue Mountains, they saw about a 15% loss. So again, not as much as white-tailed deer, but still we did lose some mule deer to this outbreak. And then unfortunately, we suspect that we lost over 70% of the Mount Hall bighorn sheep to the hemorrhagic disease virus last year. So again, it seems like pretty significant losses short term, but we don't know the long-term impacts of these outbreaks. So interestingly, these viruses are endemic in certain areas in the US. So in the Southeast, they'll have these viruses circulating every year, and they'll see some animals or whitetails die of EHD or blue tongue. When huge outbreaks occur, so the one we saw last year in Washington, those typically occur every 5 to 7 years. But unfortunately, with climate change and greater frequency of drought, we're also seeing a greater frequency in these outbreaks of hemorrhagic disease. So we speculate because we're seeing more frequent outbreaks, these populations aren't able to rebound and they're getting hit again with these viruses. So potentially greater long-term impacts than what we would imagined, a decade ago or longer. Okay, so I'm going to talk a little bit about management and very specific to Washington. So other states do similar things to what we're doing here. But it can vary, especially with CWD, because we have not detected CWD in Washington state. So the majority of our management is directed toward preventative measures. So preventing CWD from actually being transmitted to our native herds. And so some of those preventative measures are, we prohibit certain species of cervids that could be infected to be transported into Washington state. We also regulate how hunters who hunt outside of Washington, how they can bring back their harvested animals. And all of these measures are to make sure that people aren't inadvertently bringing back the infectious prion in their harvest. So we do have preventative measures in place. Obviously, we can do a lot more. So for example, in our state, we do allow feeding and baiting for the purposes of hunting. And so that is an issue that we would like to address specifically so that we can reduce the risk of transmitting CWD among individuals at those transmission hotspots. I will say we do have a CWD management plan that was adopted in December of 2021, so just last year. Within that plan, we do have a response plan outlined in the event that we do detect CWD within Washington. So we do have a plan in place if we were to detect it, and that plan basically outlines strategies so that we can prevent it from spreading to other areas within the state. So for surveillance, again, I mentioned this is incredibly important for specifically with CWD, because again, animals can survive months to years without ever being detected with the disease. And during that whole time, they're potentially spreading it to other animals. And so surveillance is incredibly important. Our agency started surveying for CWD in 1995, and that was very limited. So it was just basically if an animal presented with those clinical signs, we would test them. From 2001 to 2011, many states got federal funding. And during that time, we did expand surveillance and tested lots and lots of hundreds of animals statewide. And fortunately, all of those came back negative. After 2011, federal funds dried up. And so we had to revert back to limited testing. But I'm excited to say, as of last year, we received state legislature funds, and we are expected to conduct targeted surveillance into perpetuity. And so that's really exciting for our state, that we can now really try to find CWD if it in fact is here, or when it does appear, hopefully we'll be prepared because we've detected it at a much lower prevalence than if we were to allow it to spread. So that map that I'm showing you is basically areas that we identified that are potentially high risk because of their proximity to known cases outside of our state. So we're really focusing surveillance right now in Eastern Washington. With EHC and blue tongue, we can't really do much as far as managing hemorrhagic disease. However, we do make a commitment to monitor outbreaks. So we know that if there is an outbreak, how long is it going, what species is it affecting, and potentially what are those impacts. And so that way, if there is an outbreak, we can respond appropriately. And generally that means we adjust our harvest to reduce unnecessary take of those populations. We do a lot of public outreach. In fact, with diseases like this, like hemorrhagic disease, we rely heavily on the public to let us know when they're seeing sick or dead deer. And so this just shows you the number of reports we were getting last year. And we because of those reports, we knew that the outbreak most likely started kind of the mid to late August, and we actually had our first diagnostic confirmation of it around then. And then, as you can see, typically the outbreak peaks around mid-September, early October, which fits in perfectly with the epidemiology of this disease. So we rely heavily, because we can't be out there observing every sick and dead animal. So we rely heavily on the public to let us know when they're seeing these animals on the landscape. Okay, so just to wrap up, so what can we do about these diseases if there's no vaccine or treatment for these animals? I like to say we want to manage for resilient wild populations. So diseases are incredibly difficult to manage in especially wild populations. There's only one animal disease that's been eradicated globally to date, and that's rinderpest. And so that's a viral disease of ruminants. And it was declared eradicated in 2011. The last case was documented in 2001. But that disease had they had a vaccine that works, and they had a tool that they could actually use for that disease. Unfortunately, with CWD and hemorrhagic disease, we don't have vaccines or treatments, and so our best management practice moving forward is to prevent these diseases from spreading to new areas. And then if they do get to new areas, contain them within those infected populations so that it can't spread outside of those areas. So basically, we want to manage for healthy wild populations that are resilient to these disease outbreaks. And I put healthy in quotes because I don't mean like every individual is without any illness or is not infected with anything, because that's simply impossible. Wild animals are full of ailments. But what I mean here is that the population is healthy and managed at densities that would allow them to be resilient to when these disease outbreaks occur. Yeah, this is my last slide. Just some resources, obviously myself, but also I'll direct you to our newly adopted CWD management plan. We also frequently update our CWD webpage with information about CWD, but also our surveillance program. And then I will also direct you to, we have a just general disease page within our WDFW website that has a lot of great information about not only these diseases, but other diseases that affect Washington species primarily. So, okay, that's it.
Nice. Thank you, Malia. Thanks. And we do have a couple questions come in.
Okay.
So one on chronic wasting disease. Any study or relationship to the urine or scents that hunters typically use to mask scent and as attractants that are sold in outdoor stores contributing to the spread? Some are synthetic, some are not, some not, but not all. Maybe they would be see the symptoms before collecting or test the product for CWB.
Yeah, that's
CWD.
Yeah. Yeah, that's an excellent question. We do address this in our CWD management plan. So the use of urine-based products for hunting is an area that we identified as high risk. So those products, they could contain the infectious prion. The way they're collected is usually at captive cervid facilities, and they often batch a lot of those products together, so from multiple individuals. So unless those particular companies are testing their animals or their product, which I'll say there are some products out there that say they do test it for CWD. You have to be, be cautious of that, because there is no test that is 100% going to detect every infected product. So I will say something else that's super alarming. It's not published, I don't believe, but I've heard that individuals in Oregon tested what was supposedly synthetic product being sold. And unfortunately, they detected real urine in some of those synthetic products. And so the unfortunate part is that the production and sales of those products are not regulated, from my understanding, through anyone. And so potentially they can just put whatever labels they want on those products to make you feel better that you're using them. But yes, I would be, those products could potentially contain infectious prions.
Nice, nice. And so is there any, you mentioned that like in drought where the deer would congregate in a certain area where they're drinking, is there any forest management activities that could be done to help minimize the spread and reduce the amount of infection for any of these diseases?
Yeah, I'll just speak to, I don't know any, like, I can't point to any specific forest management, but basically, if we can manage forests and manage habitats where deer can utilize resources spread out, that we're not congregating deer. Now, deer and elk will naturally, they call it yard up, or, create larger groups during the winter time. That is natural. But if we can ensure that we're managing these populations at appropriate densities so that individuals aren't nutritionally stressed, they are able to access habitats that meet their needs, and more spread out, that's going to be our best bet to combat these diseases. So yeah, basically just managing for healthy populations, healthy densities of these populations, which also goes hand in hand with healthy forest management practices.
Nice. All right. Well, it sounds like too, definitely, since there's a need for surveillance, if foresters are out cruising timber or, driving the roads, definitely be on the lookout for any dead deer and maybe take a look at it and report it. It sounds like too, that's how they can help. So.
Yes, definitely. And I'll point everyone to, we have an online reporting system. So it's a really easy system. You just go onto our website, and if you, in our search bar, put a sick dead animal report or something like that, it will take you right to our reporting system and you can report it online. We have staff that routinely monitor it. And so if you, they'll inform all of us if they see anything suspicious or we're getting lots. Or that's what happened this last season with the EHD blue tongue outbreak. It was all of a sudden we were getting like 100 reports a day of dead animals. And so that definitely triggered to all of us, okay, we need to get field staff out and collecting samples.
Nice. All right. It looks like one last question. So did EHD originate in wild or domestic animals?
Oh, that's a great question. I don't know, and I don't know if anyone knows. The first reports in wild animals was in the late 1800s, was in the late 1800s, and white-tailed deer in the southeast. But I don't know what, I don't know if there's reports of domestic animals with a certain ailment prior to that. I'm not sure.
Yes. Okay. Great. All right. Well, I think that is it then. So if anyone else has if any other questions occur after later on, feel free to put them in the chat and we can pass it along to Malia. Thanks.
Sounds great. Thank you.
Thank you, Malia. That was a great talk. I think for our next speaker then, we're moving on to our student groups. Jean, are you able to introduce this first group? Is that what we decided, Andrea?
Yeah. So I'm not
Yes.
sure.
We're using this webcam now.
Okay, all right, yep, I'm here, yes. So as Noah is online, I believe. And yeah, so I'd like to introduce, so our students today, as opposed to what we've had the rest of the week, these students today are all part of our bachelor's programs. They're in their third year of education here. And they have completed a wildlife ecology class this last quarter. So it's actually very nice they're speaking today as they have some background in wildlife. And so this morning or this afternoon, the first speaker we have is Noah Barth. And so I will let Noah present his presentation.
Great. Yeah, thank you, Gene. Yeah, it's been really cool to see these wildlife presentations today, dealing with our habitat and wildfires and the cervid diseases. Really interesting stuff. Thank you. Let's see, am I able to share? Looks like it. Perfect. So the research that I completed last quarter, I was with one other partner named Adrian James, who unfortunately couldn't make it to this presentation today. But we looked at comparing amphibian biodiversity to kind of assess wetland health between a urban wetland that's kind of impacted by impervious surfaces and recreationists, and it's in like a suburban sort of development right next to our college. Compare that with a more rural wetland area that is off Highway 410 outside of Enumclaw, near Mud Mountain area. So this rural area is in more of a classic wetland, covered in trees, a lot of scattered pools that fluctuate in water level throughout the season. And in our urban area, it's more of a pond that retains water throughout most of the year and drains into a creek that eventually flows into the Green River. Our setup for taking these cover trap surveys was to do somewhat of a transect around the accessible southern perimeter of the pond in areas that would provide good habitat for amphibians, which includes organic material, a good substrate of mud and minimal gravel. And we tried to select places around the pond that could mimic the environment that we see out in the rural area as far as habitat characteristics go. In the rural forest out at Pat Cummins, those cover traps were placed in more of a grid form, 3x3 with one additional to make for 10 up on the furthest east side. Those characteristics also included the organic material, the adjacent to a standing water area when we laid the traps. And we wanted to see, our hypothesis initially was to hopefully find more of a diversity at the rural location compared to the urban location. But as you can see in our results here at this table, we ended up finding only two individuals of one species at Pat Cummins, and that was the Pacific tree frog or the chorus frog. We only heard its call, so they weren't even in our traps. We weren't able to trap anything underneath our cover boards at the rural area. And then compared to our urban area at the college campus, we ended up finding two different species. One of them was the long-toed salamander. We found some egg mass of about 5 individual eggs adjacent to our cover trap inside the water. And the other species we found was the Pacific tree frog. We saw 2 individuals of that species as well. So we ended up proving our hypothesis more so unfulfilled, undefined, rather than proving it wrong, because these numbers are so low. We didn't find much diversity or much count of anything really. So the main point of wanting to present this today is that to kind of discuss the constraints and implications for our research. We came to the conclusion that there's not a measurable significant difference between the two wetlands, and some things that we kind of attribute to giving us this information, this lack of data, is that the timing of the year definitely played a role, since we were kind of constrained by when the class took place over the 10 weeks from January through March. You want to survey for amphibians. We found this in our preliminary research that the best time to survey for amphibians is at the tail end of winter when spring snowmelt is underway, temperatures are warming up, and we on the ground are seeing more pools that are not frozen or snow covered so that the frogs and salamanders, all these amphibians, can find some place to set up shop and breed. So we were a little bit early in the time of year to be getting that because we found snow and permafrost still in the rural setting when we first set our traps out, which meant that 2 weeks later when we went to go check our traps, one of them had been washed away with the fluctuation of the water levels after the snow melted, and we had a lot of precipitation up in the mountains to kind of contribute to the water table down there. So unfortunately, we lost 2 traps out of our 10 in the rural area, and in the urban area, we didn't lose any traps, but we did find some disturbance. There's a trail that runs through the south side, just off the border of the pond. And we did find one of our traps was a little bit off kilter. So it could also have been messed with by hikers or curious dogs or anything like that. And the other big impact that we could see to our study is that upstream quite a ways from the area outside of Enumclaw, there was the fish fire in 2020 that burned a lot of acreage and ended up scarring a lot of the landscape. So it could lead to different water levels or different pollutants coming into this Pat Cummins wetland system. And that could have potentially impacted our findings as well. So in the future, we definitely want to pick a better time of year to complete this study. And in order to kind of rectify that difference, we left our cover boards out along these wetlands to just go check this month and next month, kind of into the end of May, early June, just to see if we end up finding more biodiversity, more species. But it won't be published as part of this research. It's just more of a curiosity thing. Yeah, so that about covers the implications and constraints about our research, and I'd be happy to answer any questions.
Nice. All right. Thank you, Noah. Nice. And so kind of why did you choose these 2 study sites?
Yeah, so both of these study areas are owned by the college campus, and they're really easy to access. Plus, they show a diverse array of kind of ground cover, forest cover, and land usage. The area out in the rural zone is just a open, minimally managed forest that takes up about I think our parcel is probably 20 acres or so, and it butts up against the land owned and operated, managed by the Army Corps of Engineers. So this area out by Mud Mountain Dam is relatively undisturbed, whereas the area up at our college campus sees a lot more action. So we were trying to compare the 2 sites based on the level of disturbance that they achieve.
Nice. Okay. Nice. And so if you were to do the study at the same time next year or so, would you change where you place the cover traps, or maybe the length of time the traps are out?
Yeah, I think the timing would be a big thing to change. Like leaving them out for another month or 2 would even be sufficient because we only had our traps up for a total of 3 weeks when we were completing these surveys. So to do it next year, if we were to do it at the same time of year, it would be really cool to keep monitoring them a couple times a month through June to see how the population levels kind of encroach into the wetland areas after a cold winter hibernation.
Nice, nice. And so did you have any issues doing the field work, like, placing the traps, or did you have to readjust where you were placing them depending upon the site conditions?
Absolutely, yeah. We ended up needing to break the ground up a little bit with the Pulaski at the rural area because it was a little frozen and had about half an inch of snow over the top of it. And we ended up placing the traps in areas that looked like they were elevated out of the water table. But when we went back to check on them 2 weeks later, a few of them had flooded over and we couldn't find the cover boards again. So that would be another thing to change in the future, is to kind of monitor more closely where the water level is going to rise to before we place traps that would end up being flooded. But our other traps are in still really good locations that didn't see any flooding, and that still on paper should be characteristic amphibian habitat. And the sources that we had for determining like areas to place and kind of the timing of the season came from the EPA and the US Forest Service as well, discussing different wetland management kind of health assessments. And there was a lot of reading. So this research paper that we ended up turning in turned out to be more of a literature review based on those things, since we didn't find adequate results.
Nice.
But it was fun. It was interesting.
Nice. And kind of what amphibians would you have expected to see there? Or maybe if you were to go in a different time of year, what amphibians would you find?
There's definitely Northwest salamanders, and we've seen evidence of giant Pacific salamanders out in the Pat Cummins area near Mud Mountain Dam. Plenty of Pacific tree frogs and western toads are in both of these areas as well. Some rough-skinned newts and alligator lizards I wouldn't be surprised to see out there either. But yeah, unfortunately we only found the Pacific tree frogs, which are still really, really cool animals to find, and the long-toed salamander egg mass.
Nice. Okay. Nice. All right. Well, does anyone else have any last questions for Noah? No. All right. Well, thank you.
Yeah, thanks, Noah. Appreciate it. All right, and I think now we're ready for our second Green River College presentation. Perfect.
Yeah, great presentation. So thank you, Noah. Yes, we have another presentation from two other of our students, bachelor students. We have Raina Stokes and also Tanner Larson. And they were in that same class and talking about wildlife. So they've got a great presentation to share with you today as well. So
Awesome.
take it away.
Thanks, Jean.
All right.
Right.
Welcome.
Let's hopefully get a PowerPoint running up here.
We've got two monitors, so give us just a second to actually figure it
We
out.
made a little PowerPoint. And it's probably just going to be a bare screen. Does it look good? Are we seeing a PowerPoint?
You guys look good.
Awesome,
Awesome.
thanks. Like Jean said, my name is Raina,
And I'm Tanner.
and we are third-year students studying forest resource management here at Green River. And like Jean mentioned, we got we had the opportunity last quarter to study wildlife ecology, which was super cool. We were tasked with conducting an exploratory study, or like Tanner, or like Noah mentioned, a literature review / experiment that was centered around wildlife. With limited time in the quarter and an interest in restoration, Tanner and I wanted to look at what wildlife could tell us about different restoration techniques. We both have a background in riparian restoration, which gave us access to some pretty unique wetlands with very, very different management styles.
And that's why we constructed our study around the differences in amphibian diversity with a focus on natural and restored wetlands in the Pacific Northwest. Our question that we had was structured around studies based on benthic macroinvertebrates. And so we kind of used the similar basis, saying, is there a notable difference in amphibian species found between a restored wetland and a historic wetland? And from these results, do these restored wetlands hold the same ecological value? Using that benthic macroinvertebrate kind of analysis as our backbone, we hope to kind of use their assumed trends to kind of strengthen establish our hypothesis.
Yeah.
Which is, given that the historical systems have had time to establish, we believe their wetland will have higher abundance of amphibian diversity, species richness, and evenness.
And this photo here is a picture of the historic wetland that we did work in. Jumping kind of from where Tanner was saying, this idea started with studies that we had done with macroinvertebrates. So we wanted to learn a little bit more about amphibians. Amphibians are a super, super unique, complex class of animal because they have multiple phases of life where they experience super dramatic physiological and morphological changes. So throughout their life, they'll utilize wetlands, riparian habitats, and the upland forest zones. It's during their breeding season that they use lentic waters, like what we find in the wetlands, for their breeding. And then as the amphibians mature and kind of morph, they'll typically move into the upper forested habitats for the remainder of their life. And it's in that adult form that the amphibian skin is super thin and permeable, which allows them to fluctuate between those two ecozones, the aquatic and terrestrial.
And that's why, considering amphibians, we have to look at them kind of as a nutrient bridge between these two critical ecozones. Throughout their larva stage, they're collecting nutrients that's often unavailable for most upland predators and apex predators. And so they are valuable in pretty much nutrients distribution. They are complex, and they are a direct link to trophic cascades between wetlands and uplands. Many amphibian species in Washington hold a unique trophic position as a mid-level consumer. As you can see kind of from our little diagram here. It is also good to note that although amphibians are a sensitive species, they are highly adaptable, and most species can survive in severely degraded land. Many of the amphibians are an R-type species, which meaning that they primarily focus on reproduction as their means to survival, which also can aid into possible mutations that can kind of allow them to take into an environment.
So amphibians were just the perfect thing to study, basically.
Learn that they're awesome.
Here you can see a big old map, not a great map, but here's kind of the Puget Sound or the Salish Sea, if you're familiar with the area. Our college is kind of smack dab in the middle of this map. And we wanted to look at two sites, kind of in the upper reaches of the watershed where there wasn't as much disturbance, and then lower in the reaches of the watershed where there was some more disturbance. So we chose two sites that had very different styles of management, but actually were classified via the DNR as a similar type wetland. So both of the sites that we chose were 1/2 acre with very, very similar ecologies, but we tried to highlight in this slide the difference that they actually, the differences in how they look on the ground. You can see in the upper reaches of the watershed at our control site, there is a bunch of trees and lush forest and no disturbance. And then in the top photo at our manipulated site, which is lower in the watershed, it is literally consumed by urban sprawl. So Tanner knows a bit more about these sites and he'll talk about them some more.
Zooming in just a little bit more, Pat Cummins is a Green River College kind of owned area. It is naturally regenerated. It's a mature working forest and it's utilized by us for educational purposes. It was pretty much the ideal location for kind of surveying these because it is a mosaic of wetland typings. Through the WDFW typings, we have emergent, we have freshwater, we have shrubland. It's pretty much everything. Under the DNR, we have type A and forested wetlands here. Just an amazing amalgamation. Smith site is a recently restored wetland with ongoing maintenance. Being in an urban environment, this property's had multiple owners with differing management objectives. And so you kind of get a conglomerate of just messy practices. Really rough there. Urbanization has created pretty heavy sediment deposits going through the area, which has altered the morphology of the land. And the impervious surfaces that were caused, granted to like road making and all that, has actually added to the amount of water influx and that the stream system gets, which makes it a little bit more prone to flooding and has actually channelized the stream. This entire urbanization period has introduced a lot of invasive vegetation, and the whole area is pretty much, or was before 2018, it was entirely reed canary grass and blackberry. The property has been designated as a wetland for the past 40 years, but has had literally no human interaction in terms of restoration. It has just been a victim of urbanization. But to kind of make these both relate a little bit more, we made sure both sites had a fish-bearing stream running north to south, and that both of them were leading into water sources that would eventually drain into the Puget Sound.
Now we get to the cool stuff, which is like actually starting to conduct or like theorize how we're going to do our study. So we picked our locations and then we dove into some further research trying to figure out what kind of amphibians actually utilize these systems. And we found 8 native ones that use wetlands, and of those 8, we expected to find 4.
We expected to find northwestern salamanders, long-toed salamanders, Pacific tree frogs, and red-legged tree frogs, just because of the timing. Northern red-legged, that's my bad. Northern red-legged frogs, just due to the timing of our survey and just because of the previous kind of heat that we had. We expected to also see signs of invasive bullfrog just because they're so predominant here.
Yeah. So here is our methodology. This was a lot. Noah kind of touched on it in the last presentation. Our quarters are 10 weeks long, and so there was quite a lot of research that had to go into this before we were actually able to get boots on the ground and conduct these surveys. So kind of going back in time, it's February, and we are considering that there are a ton of different types of surveying methods used for amphibian monitoring. And each one of these methods is advantageous for different species, different ecosystems, and different times of year. So because ours were going to be occurring in late February during breeding season, we wanted to try to capture both egg masses in the water, but also the potential breeding adult or traveling adult that's migrating through different ecozones. So we decided to utilize a system called time-constrained surveys, which is a method that minimizes impact to habitat and species, and also increases our chances of seeing a species because we have the, like written into the methodology, we have the right to choose where we look within our plot. So time-constrained surveys, they're systematically placed throughout a study area, and you basically go from plot center to plot center scanning for amphibians visually. These surveys are super helpful for providing or for determining presence and absence of a species, and for providing insight on species-specific microhabitat choice, which in this experiment we're looking for potentially 8, and each one of those species has a different microhabitat choice.
And between these sites, we specifically tried to constrain our area within a type A wetland and made sure that each of our plots were in a ½ acre. Each plot contained 30 study plots with measuring 25 feet by 25 feet. And these plots were chosen because they were small enough for us to get through it in a 10-minute time period between each survey.
Yeah.
And based on the recommendations of the EPA, we found that the ideal kind of timing and weather was early-ish in the morning and during or after a little bit of light rain. So we kind of set our time within 2 consecutive days at 10 a.m. After where weather patterns was suggesting that light rain was going to happen. Surveys were conducted by walking the median of each point, stopping at the survey center, and then conducting an analysis there. We were specifically targeting places that we deemed ideal habitats, such as coarse woody debris, stuff like that. And if an egg mass was sighted or species within the plot, they would be recorded as a sighting. And pretty much that's how we rolled. At the restored site, we found 1 Pacific tree frog mass, and it was connected to woody debris roughly 2 feet underwater. Given this habitat, given the habitat availability from my previous work at the site, I actually expected a lot more long-toed salamanders, just because whenever we were digging holes to replant in the site, we found long-toed salamanders just crawling out. So we were just expecting to see them very prolific here. We also expected to see invasive bullfrogs, which luckily we didn't, which is kind of a win. Kind of diving into what we found, the Pacific tree frog, they're a resilient species, prolific breeder, who have been found to lay egg masses in sources as insignificant of 4-inch deep puddles. They have a large range going through the entirety of Washington, and they also have been known to not even be near water sources. You can find these guys in woodlands, forests, meadows, but during breeding season, it is most likely you'll find them at a wetland. That's what you hear all the time. They are loud. They have a pretty long breeding season, which can start as early as November and span through July. And due to their abundance in the Puget Sound, we can't really use them as an ideal candidate for indication of wetland quality. But there's some other species like the red-legged that do. So let's hop into that.
Yeah, so at our historic site, we expected the species that you see here in the graph. We did not find all of those species. We did only find 1 unique species, that was the northern red-legged frog, but we did have a little bit more success because we found 2 egg masses instead of just 1 egg mass. There was no presence of salamanders, no other frog species, which was surprising. And then the small win for this site was no invasive bullfrogs. Diving into the habitat description of the red-legged frog, just to kind of compare to the Pacific tree frog, the red-legged frogs are much more niche. They're found primarily on the west side of the Cascade Mountains in Washington. And they're going to kind of gravitate towards much more private or slower environments, unlike the Pacific tree frogs. So these guys want to spend their time in permanent, quiet, forested wetlands. And then for depositing their eggs, they're gonna have even tighter parameters, and they're gonna want a really, really quiet lentic wetland with deep water to deposit their eggs. They, excuse me, sorry about that. They deposit their eggs in a quiet lentic environment, and then after that, they will migrate upland for their post-breeding interval. And that's pretty cool. They'll actually migrate super far into their forest ecosystem. So they're a really good niche animal to use as an indicator for habitat fragmentation. And then just to put in there, I feel like we were super lucky in our survey to find this species because they have a very narrow breeding window that lasts between January and April. So it was a big win.
Big win. So for our results, is there a notable difference in amphibian species found between a restored wetland and historic wetland? Unfortunately, we don't have enough data to really do anything with. And so our following point, do restored and constructed wetlands hold the same ecological value? We just don't have enough data. But one thing that we can pull out of this is, although this might not be sufficient under scientific analysis, our data observed still has potential to convey current habitat types for these early breeding species.
So moving forward, we want to conduct these studies in a way that actually makes sense and is beneficial to the land. So we do recognize that there were some flaws in our study.
In the process of identifying these ideal wetlands to compare, our initial mapping projections led us to believe our control site, Pat Cummins, was a type A wetland, but it was not delineated as such. We made the mistake of thinking the WDFW map was the DNR. We had a mix up pretty much. And so under the WDFW map, though, both sites are listed under freshwater or shrub wetland, which kind of still gives us a bearing for a control site. And although these amphibians can tell us a lot about habitat, there is research that suggests that urban environments are far too variable for these species to be utilized as, as indicators. Thank you. And mainly it is just due to their adaptability. There's also studies that suggest these little guys cannot be used in general for indicator species because they don't match proper criteria. Those are always conflicting studies that we have to kind of utilize during our research.
In addition to like research-based flaws, as we keep harping on in this presentation, there was limited time to study or to conduct the study. And so that definitely narrowed us down in our analysis. And then we also wanted to notice that there were definite unusual weather patterns that could have correlated to our low observation findings. Like Noah mentioned in the previous study, there was snow this year, kind of in the late winter, and then additionally last year, there were some pretty intense heat waves in the spring, which was last year's breeding season. So kind of taking all of these flaws and hoping that we can put out better surveys in the future, we believe that if timing survey is adjusted to late March, we may have had more success observing these larger niche target indicator species.
And also on top of that, we saw both of our findings in deeper water. So our study area really didn't have much of that. So future surveys may benefit from canoe or kayak travel to kind of see those deeper waters. And the reason why we're suggesting something that's not waders is just because every step we took, we just made a cloud, and it took like 6 minutes for that to dissolve before we could actually do our survey.
Yep, very true. So with time permitted, we conducted one survey in each one of these areas, and we really believe that we need at least 5 more surveys in each one of these areas throughout the breeding season to give us some quantifiable data that we could do some analysis with.
And again, if this was to be done again, water quality tests that can gauge the parameters and benthic macroinvertebrate surveys that can identify pollution could also be utilized just as kind of a building block for this idea of research.
This is our last slide, just to kind of wrap it up. We did talk a lot about these discussion elements in the flaws, but Tanner will kick us off with some.
I found, so just going off of that mapping idea, I found like, I understand as a group of foresters, we all host diverse land management objectives. And so as a researcher, we kind of have to look through those delineations to make sure we're pretty much finding the best one that caters to the amphibians in this type of experiment. And so I guess what I'm trying to say is differences in agency goals lead to discrepancies in wetland delineation. So how can we as land managers identify our best capabilities to cater to these amphibians?
Basically, it's super important to get out on the ground and verify all of these wetlands. And I think that was something we learned in this experiment, was you need to get out there, you need to verify everything. But I want to kind of discuss the wildlife oscillations that I touched on. So last summer, when we had those big heat waves in the early spring, we predict that those could have heavily impacted the egg masses that were laid back then. These oscillations that wildlife populations have are totally normal. But when humans are impacting an area like the the study area we had in the lower reaches of the watershed. We want to understand how the amphibians are using this space and when they're using this space so that we can be better land managers in the future.
And also in addition, to like kind of wrap it up, we need, or as when we were out there, we thought, like when we initially went out there, we assumed like climate or habitat fragmentation and habitat loss was their biggest concern. But we kind of discovered that it could just be, or weather patterns, climate change is also a really big threat, especially on the west of the Cascades, because these guys are used to a specific parameter that I don't think we've hit pretty consistently over the past few years. And that pretty much wraps up our exploratory survey. This was a beautiful picture Raina found on one of her hikes.
Yes. Do we have any questions or comments? We went a little long, sorry about that.
No, not a problem. Good. Nice. And so for the urban wetland, kind of when was that created?
That was the urban wetland was actually, from what our research shows, was tradition or like historically a type A wetland.
Okay.
Tanner had the opportunity to locate the property as a degraded site, and he worked with the landowner to like restore it. So he's still actively working and managing it.
Cool.
I go off. It's so originally it has been designated as a type A wetland for the past 60 years. It actually was, so the landowner that I've been able to meet pretty closely now, because we work pretty much on his property to restore it. He has been told by Kent pretty much don't touch. And so when we got there, it was blackberry, reed, canary grass, all sorts of the lovely invasives that you would expect.
Perched culvert,
yeah, even lovely stuff like that. And so when we got out there and we started putting together the project and replanting and stuff like that, there was a large water event that just kind of pushed all of the blackberry canes that we kind of made and pretty much opened up the wetland that we, in a way we didn't expect. And so since 2018, over the past 4 years, we've been able to see kind of more plants come back. We've had Douglas spirea just taking off now, which is amazing, which to me, when I see spirea, I think of a wetland immediately. So, and the wildlife there just seems to keep blowing up. So we use that as kind of our base, just because there is a lot of similarities, just unfortunate fragmentation from urbanization.
Nice. Now it's amazing what happens when you start restoring a site, what all the natives start coming back once you give them that space.
Yeah, elderberry. We didn't even see elderberry at first until, and now it's everywhere on the site. So
nice. Yes. And so did you run into any issues doing field collection? Did people come by and, and ask what you were doing if you were visible to the public or?
We were definitely, because our sites were both private property, we did not run into anybody that I can remember.
The landowners. I think at the very end when we left, we got caught in a half-hour conversation with them.
Okay.
I think the most difficult thing was actually, we were super ambitious with how thorough we were going to search each one of our plots. So when we first wrote out our methodology, we wanted to spend 20 minutes in each plot, which for 60 plots overall was absolutely unfeasible for us as students. And so we had to narrow that down. But I think we both would have wanted to spend more time studying, like out there surveying. And so that was kind of a blessing and a curse, I guess.
By the time we actually got to do our research, we had 2 weeks left of school pretty much.
Okay.
So we were running on fumes.
Nice. Nice. Well, does anyone have any other questions at all? Nice too. So looking ahead, you kind of already mentioned this, but so it sounds like if you were going to do the survey again, you would do it at different times and maybe collect other data or join with another project for them to collect the water quality portion of it.
Which is super, I'm glad that you brought that up, because this year specifically, and you guys have maybe saw some of the presentations, a bunch of the first year students ended up doing a lot of their research at that Pat Cummins site too, where they were looking at macroinvertebrates or water quality. And so there is a lot of opportunity there for collaboration between like bachelor students and associate students. And I think that could really, blast our research off.
I think it'd be fun. Yeah, absolutely.
Nice. Cool. No, collaboration is always good.
Yeah, for sure.
Okay. Okay. All right. I think that's it. We'll turn it over to you, Elijah. Thanks.
Thank
All
you
right.
guys.
Well, thank you. That was actually a really excellent presentation. Impressed. And I want to just thank everyone again, everyone for showing up today, and our speakers. This is it. This is the end of our conference for this week. I appreciate everyone who's shown up, all the speakers over the last 5 days. Again, my thanks to our sponsors for making this possible, and to the Northwest office and the planning committee. You know, this really came together, I think, and I hope everyone enjoyed it. It was really educational for me. In the next week or two, hopefully we'll be making some materials available. You guys may receive a survey. If you have any questions or comments or anything else, please send them to myself or to the Northwest office. I think those emails are provided in the program. Otherwise, make sure if you've got looking for credits, Andrea's posted that in the chat, so follow that link, use the information to sign in. And otherwise, Andrea, is there anything I'm missing that you want to add?
Nope, I think we're good. And hopefully if, everything stays, if there's no more variants that come up, we'll be able to meet in person next year. So.
Yes, yeah, looking forward to that. So thanks again, everyone, and enjoy your weekend.
Okay, thanks.
Oh, you got a hand raised. Or no, is that clapping?
I think Jean's clapping. So.
Okay. All right. Everyone enjoy your weekend. Thanks so much for showing up. I appreciate it.